To start off with, let me just apologise for the state of my handwriting in a lot of these slides. I'm not quite sure what happened there. So we're going to be talking about receptors, and there are two types we're going to be talking about, which are your extracellular, which is when they're on the surface of a cell, so sort of in the plasma membrane and then sticking out of it, and then intracell receptors, which are often types of enzymes or proteins found in the cytosol or nucleus, or they can sort of move from the cytosol into the nucleus and like move about a bit. They can also act as transcription factors. I'll talk about three things that receptors can sort of trigger, and these are metabolic enzymes, which can obviously alter your metabolism. You've got cytoskeleton proteins, which alter the shape and function of cells, and you've got transcription regulators, which alter gene expression. And we'll talk about the three classes of receptors. First up, you've got your G protein coupled receptors. G protein acts sort of like an on and off switch. It's classed as on when it's associated with GTP and off when it's associated with GDP. Sounds very similar to your ATP and your ADP, so just sort of think about it like that. But instead of adenosine triphosphate, it stands for guanosine 5-diphosphate or guanosine 5-triphosphate. And I'm so proud of this little diagram. So on your left, you've got your ATP pathway, and on your right, you've got your GTP pathway. So you can see it's very similar, there's just some slight differences. And a tip my lecturer told me was to think of them as different types of currency. So they do the same thing, they just got different letters. And now I'm recording this, I realise maybe why my handwriting was so bad at the beginning, because I'm so proud of these diagrams. My sort of steady-handedness was used up in trying to make these rather than the actual words. So what is a G protein coupled receptor? These are types of proteins that belong to the 7-pass transmembrane family. That means the protein sort of goes through the membrane 7 times. So you can see it took so many times. I was an idiot and I didn't put it through 7 times. And when a ligand binds to that, it causes a conformational change and activates the G protein, which is also near the membrane. So your G protein is made up of 3 subunits. You've got your alpha, beta and gamma subunits, and your alpha units associated with the GD or the GTP molecule. So when it's inactive, it has a GDP on it, and when it's activated, it has the GTP on it. And depending on the type of G protein, the subunits can either stay together or separate apart. So you just have your alpha unit and then your beta gamma complex. And this activated alpha subunit can then cause like further activation downstream. So, for example, it can bind to a target protein in the membrane, which activates it, and then after time, the ATP is actually hydrolyzed back to the ADP, and the protein becomes inactive as the alpha subunit becomes inactive. It then reassociates with the beta-gamma complex, and it will remain inactive until a new ligand binds and the signal starts over again. And then the next one's going to be an example of how the beta-gamma complex can actually activate a protein. So as the alpha subunit's active, so is the complex. That can go and activate a protein downstream. For example, here we've got a channel protein. That can activate it, so it opens, it allows molecules to move through. Then when the signal stops, such as when the ligand dissociates, the alpha unit becomes inactive again, rejoins with the beta-gamma complex, and then the whole G protein's inactive. So whilst we're talking about receptors and ligands and all that type of stuff, let's talk about some adrenaline. So adrenaline's a hormone which is caused by stress. So stress causes an increased regulation of the sympathetic nervous system and causes the adrenal gland to secrete this adrenaline. Your adrenal glands are like a little bit above your kidneys. You may also hear it being called an acute stress response, and I'll give you some examples of symptoms or things that occur when adrenaline is released. So you've got tunnel vision, sweating, bladder release, slow digestion, dry mouth, shaking, increased heart rate and a flushed face. This is because all your energy is sort of being diverted to running away or fighting for your life. So you're not really going to think about swallowing or think about digesting digesting and you're not going to like consciously control not going to the toilet. But if you look at it at a molecular level, you'll see that it's actually your glycogen and fat in your muscles is being released so that they can be used in respiration. This allows for faster responses so that you can fight or flight. And it's all mediated by the androgenic receptor, which is a type of G protein coupled receptor in the CAMP pathway, which I'll go on to in a second because I need to go over some key terms. So you've got kinase, which means the addition of a phosphate from an ATP molecule to a protein. You've got phosphorylase, which is the addition of an inorganic phosphate to a protein. These both make it active, but then you have phosphatase, which means the removal of a phosphate from a protein, meaning it's made inactive. I know that may look really mind-blowingly complicated, but I'm going to go through the steps with you. This is the CAMP pathway. First up, you've got your adrenaline attaching to the receptor. This activates the GPCR, which then activates adenyl cyclase. This causes ATP to convert to CAMP, which is cyclic AMP. This then activates pKa, which is called protein kinase A, which then activates phosphorylase kinase. Your phosphorylase kinase then activates your glycogen phosphorylase, and this all results in glycogen breakdown. So there is an image coming up next just of the stages, so feel free to pause on that and read over it. Again, probably not as neat as my drawing. And this bit is just sort of an image of the process of making ATP to CAMP to AMP. We were told we didn't need to know it, but I thought it might be nice to just sort of have a look at. Because when your CAMP concentration increases too much, it's actually converted into the AMP by phosphodiesterase. This cycle is also very important in transcription regulation, as your pKa can move into the nucleus, as I'll show you in a second. You've got your same process. Your adrenaline attaches to the receptor. This activates the G protein coupled receptor, which activates your adenyl cyclase, which causes conversion of ATP to CAMP. This activates your protein kinase A. Your activated protein kinase A then actually phosphorylates, aka activating a transcription factor, which then activates a target gene ready for transcription, which forms your mRNA, which is then used in translation. It could also be used to stop activation of target genes, so that means the mRNA isn't made and there's less of that protein made, so it in a sense sort of turning it off. So you can sort of either have transcription factors that either turn on transcription, turn off transcription, or enhance transcription, so you get more of an already activated gene being transcribed. So I'll leave this video here. I've still got 2 more types of receptors that I need to cover, but this video is already quite long, so I'll do them in separate ones. So hopefully my handwriting wasn't too bad for you, and I'll see you in the next ones.